And record.
Given the number of times I've been asked to do like podcasts and stuff, especially because of like COVID and bats being right now, it's like pretty normal for now. It's like everybody's asking for commentary or, you know, something related to bats right now. So it's been a stressful few weeks as that too, because so we have this global bat network that's like the network of all the networks. And we were supposed to be waiting to see if we get our NSF grant to start it so we can do the activities for it. Except then this happened. And so some of our, I'm a co-PI on it. And in our executive committee, they're like, so maybe we should start this earlier because we kind of need it like right now to address this emergency issue.
I know.
And so I'm like, oh shit. So suddenly they're like, we have to do all this work with no funding now and see what happens.
Yeah, like the long overarching reason for the project, or like a partial justification for the project has just happened. And now like
basically, no, it's basically like the formation of it has been discussed since 2000 and I want to say 13, or like something around there, like 2013, 14, and then they talked about it in South Africa, and we had the last international meeting before Thailand also. So it's been like 4 or 5 years that they've been talking about it. And then finally they were like, okay, we're going to apply for this funding and it's going to happen. But then what happened was, you know, the funding is still like slowly going through the pipeline because it's, you know, the government. So now we can do the stuff that coalesces all of us into a network. But some of the activities that we had planned were having these student-based projects. So we're going to have later stage PhD students to maybe early postdocs, and they're going to form these working groups to tackle more integrative questions that you can't answer without cross-pollination of different disciplines and different regions for you know, different kinds of bats.
Like what's an example of a really integrative question? Oh, sorry. Yeah, keep going. Yeah.
So, well, an example of that is like if you look at ecoevolutionary dynamics, for instance, like with fruit bats, you're going to get really different dynamics for something that's a fruit bat in the neotropics that evolved from phylostomids compared to the fruit bats that are in the old world tropics, which are all in the family Pteropodid. Because the phylostomed fruit bats are a smaller clade within the phylostomids. That's like the super diverse family that has all the different eating things.
Right, all the different trophic strategies. Yeah.
Yeah. So like every way you can get food is in that family, which is why they also have all this other stuff.
Yeah.
But then you have this big family of just fruit bats that are pretty like convergent in terms of most of their, you know, morphology. And these like phyllostomic guys are like, we have cool nose leaves, we have like long skulls, we have long tongues, and you're just like, what the hell, man? Like,
what are y'all doing?
They can look bad over here in the whole world.
Oh, come on! Everyone likes fruit bats. Everyone likes flying foxes.
Oh, no, our bats are cuter for sure. Yeah. But like, they got all the other like cool evolutionary adaptations.
Yeah.
So, you know, puts us to sleep.
They seem to have a little bit more skull evolvability, maybe.
Yeah, so we would have students that would be from these different regional networks that can contribute because they know about the bats, but then we also have people who would be from the morphology networks and the genomics networks. And it's the same kind of thing with like disease ecology. We can get people who are actually from like a physiology background and then like a genomics disease ecology background, and then also people who work in different areas like tropical bats versus temperate bats. So you're gonna get these students that can come together and form working groups, about 4 or 5 students. And we have funding that was attached to each of those working groups so that they can, you know, address these bigger questions. And we had, I think, 3 or 4 of them suggested in our NSF grant that were like really interesting ones that are about, you know, rules of life stuff that NSF loves. But, you know, now we're like, well, we're going to start all the network stuff. And then like, if they fund us, I guess we'll be able to do the project stuff, but we need to do the other shit like right now.
God. Yeah.
It's been like a crazy time for anybody that works on bats. I feel like because of that. I think everybody's been scrambling because like every single country that has a lot of bats that are in markets or like in roosts that are really close to people, their immediate reaction is just to say like, oh, we're just gonna cull all of them.
Yeah, we should probably kill all of them, right? Because they cause diseases.
Yeah, and they're just like,
we should just kill every bat.
It's like the dumbest thing. And they had a study a few years back that showed that they culled them in Peru, the vampire bats, and it was to control rabies. And what happened was, if you do that, you're gonna locally solve the problem. But then you have an empty niche space, and then there's gonna be bats coming in from other places, and it just replaces it. But there's also like, you're taking out all the adults that grew up and they're tolerant and stuff, you know. So like, there's all these other ecodynamics that are going on, and you didn't think of it basically. And so you actually have higher incidence of rabies after they did the culling.
Oh, no way. Because it was kind of a sink. It was like, they were just
and then you had
in.
all these adults that aren't gone, and they've seen this in other animals also. So like in the UK, they culled badgers because they were afraid that they had some, I forget what it was, like something. They didn't
They
like
called
them
what?
also because they thought they were like badgers in the UK.
Badgers? What?
Yeah, this is a thing a couple of years ago, for real. So like, they culled them, and then people started like, you know, doing monitoring of them after this cull, and they were like, hey, the amount of like diseases in them is higher now after you killed all the ones you caught last time. And they didn't even think of this in the beginning. The reason that they killed them was because they were like, oh, they're annoying and digging holes in everybody's backyard, you know? Right. And like some dumb reason. And then now they're like, oh, now we have a serious like wildlife health issue. And you're like, oh, well, we told you not to kill them in the first place.
Yeah, suddenly the place that you kill everything becomes like a travel hub.
Yeah, because everybody's like, oh, free space. Yeah, sure, let's go there, right? So yeah, this is like literally everybody who works on like wildlife disease, like, this has happened before. This isn't new to not even just bats. It's like to other animals also, you know? Yeah. So yeah, and another really cool evolutionary thing, well, not cool, but interesting, is that you look at all the animals where they're seeing potential spillback. So, you know, if a human has it and then it can go back into a different animal or a bat even, like with the tiger in the Bronx Zoo. So they see that in cats and in ferrets based on what we knew from the previous SARS, right? And if you look at the other animals that they've been testing, that they said that it can go back even if it becomes just like asymptomatic or like a dead end host. It's all the animals that belong to a lower area, which is like the big group of mammals that are in the, you know, northern supercontinent, the Gondwana,
the Eurasia.
So you're gonna get that's why you're gonna get bats and carnivores and ungulates all mixed up in that, because that's all your you know, big guys who are able to compete and like outcompete the Gondwanan ones. Well, guess what? Like, all of them have large reservoirs of diseases because they've been building them up for millions of fucking years. No surprise that like normally they have them, and then now that it's able to spill back on them, even if it doesn't kill them, they're gonna still get it. And you're like, oh, well, that was dumb that we didn't think of that to study that more. So that's why everyone's scrambling to sequence as many things as they can right now, because now they're trying to say like, okay, wild species are gonna get this spill back, because we have no idea what the background virome like looks like in general for most environments, but it's so bad for particular ones, especially because like people only pay attention to like domestic animals, bats, and like dogs sometimes in certain places because of rabies. But, like, no one really looks at, you know, any other animal, and especially the ones that you find in like a wildlife market. Nobody really does that kind of work because all the money's for the conservation of them and not this disease dynamic. So yeah, there's a lot of like weird stuff going on right now with the ways that you can think of evolution.
Yeah. So getting to bats and the virome, like, do bats, do you think they really do have more viruses than other mammals because they're kind of facultatively gregarious and they disperse a lot? And
no, so there was a paper that came out last week. It was in PNAS, I think, and it was like a study that showed that basically bats have more viruses because there's more species of bats. And that's also true of rodents, and that's also true of birds, you know? And so they looked at birds and mammals, and they showed that the more species rich your group is, the more viruses you're gonna find in them. But also there's just the fact that we've been like dumping research effort into bats for a while. So like they control the species, but you can still find that bats have more. However, there are elements to that which the mechanisms are different between the different organisms, but it's not like it's like the bat's fault, you know? It's they just have more because there's more species of them. This is what happens when you evolve. It's just a normal
shouldn't have diversified so much, bats.
Yeah, it's like mutations accumulate and you expect the center of evolution to have more species, right? It's like the same
Yeah.
kind of premise. And so like that's not that surprising because they can
Hmm.
tolerate it too. So like
Right.
evolutionarily, the mechanism is like, by species number, that's why, but like mechanistically, it's partially because they can fly.
Hmm.
What is why people think that they can tolerate so many viruses, existing in their physiology. So basically when you fly, you use a lot of energy. And when you use a lot of energy, your internal body parts is like just shedding off these free radicals into your bloodstream.
Oh, gotcha.
And so because there's so many free radicals, if your body was trying to just respond to it and you're a bat, that's like too much immune response. You're going to get that whole like cascading thing that's causing people to crash really hard right now for COVID
Hmm.
because of the anesthesia.
Yeah.
And so you don't want that. And so the bats have this kind of like half immune response.
Hmm.
And they kind of tamp down this whole thing so that it's all tolerant within their bloodstream. So the viruses just hang out because they don't get like fully killed. And
Huh.
because of that, they just have built up a lot of evolutionarily, existent like things like that. And birds have
Yeah.
the same issue also, right? So like it's a flight specific thing for bats because no other mammals fly.
Hmm.
And so that's the working hypothesis right now for
That's interesting.
why they have a mechanism that makes it more likely, right? For
Could it
rodents, I would
partially
think
be
it's
like
because they.
a body size thing too, because you just have higher metabolism if you're smaller body size, or?
No, because so bats actually their metabolism is high in the sense that they need a lot of kinetic energy and metabolic energy process in order to fly because it's
Right.
costly. But if you're looking at like the body size to energy cost thing, they're really different from rodents based on body size.
Hmm.
And you look at those allometric curves, you'll see like rodents at the bottom and like elephants up here, and it's
Right.
like this, right? The bats are like over here because the
Oh,
bats do
huh.
not follow this curve because they're using a ton of extra energy to fly. So they're actually expending more energy than you expect for their body size.
Gotcha.
So you're getting a lot of things that can also not really predict how they're actually doing things like energetically because we base our flight models on birds.
Hmm.
So bird wings are straight and they have feathers coming off them, right? So it's a fixed wing, and that's how our airplanes are built too. But bats, their wings are their hands, and so they do this, right? Because it's the long fingers.
Yeah.
And what happens is if you try to divide a wing to like energetic costs, it's based on these rectangles that you draw along the wing. And it
Hmm.
works for a fixed wing pattern because you just distribute load equally. But that doesn't work if you're doing this with your hand
Oh, no
because then you
way.
overestimate the amount of energy you actually need. And so like no one for the longest time could explain how does a flying fox feed themselves because they would need to eat like 2 tons of fruit to get the amount of energy to fly when they're like 2 kilos because like their wings do
Yeah.
this and their
Right.
wings like 6 feet, right? So like how the hell does that happen, right? And so they couldn't really calculate it correctly with the way we do fixed wing patterns that they knew was wrong.
Hmm.
And about like 10 years ago-ish was kind of when we started like mathing this out better. And they were like, we're going to draw like trapezoids into the area where the parts of the wings are.
Kind of
And that
between the
actually
fingers.
explains it better.
Oh, cool.
And so like, oh, that makes so much more sense now. Cause it's like, that's how it bends then, like, duh. Right. And
That's really
so
cool.
that's why you're seeing all these places that now can make like robots and stuff of them because we're starting to figure out how they actually fly.
Yeah.
And we like honestly like for a long time the very basic question of how does a bat really fly, we didn't really know that very well. Like we knew at a very basic level and just like
Right.
no one said anything.
Flapping
So
is
yeah,
involved, I'm pretty sure.
yeah, it's like there's some flapping going on and like profit happens after. I don't know
Yeah.
what's the
Yeah.
step, but like something happens and everyone's like,
Mhm.
yeah, flying, cool.
Question mark, question mark, question mark, food. Yeah.
Yeah, that's all we need to get to, right? Is like somehow we get to the end and we don't bump into a wall and we also can eat, right? So like bats are happy.
Yeah.
But yeah, there's a lot of weird like really basic questions that we have no idea about. And like I'm coming from like a bird background originally when I was an undergrad.
Oh,
So
okay.
so we studied really specific things like the sexual selection of this one species and how
Hmm.
the females are using honest signaling from the males in order to do mate selection.
Hmm.
And in the case of the species I was studying, the common yellow throat, the males have a lot of ornamentation and they have also like monogamous pairing, but a lot of the experienced males actually get most of their offspring from extra pair mating.
Okay.
And so we didn't really understand like how females can figure out which males are more fit in order to do the extra pair mating, and like which signals are like honest for that. So like that is really detailed, right? Whereas like if I told you, okay, most people who study bats don't study sexual selection because they're like, oh, they're blind. So like, who the fuck cares, right? And I'm like, mine aren't blind, and there's a lot of them. And guess what? We know they have leks, so there clearly is sexual selection going on. And we're like, how do they do it? And like, everyone's like, I don't know. That's like they form leks, and then like question mark. Everything else is like, I don't know. Stuff happens. That's like
What do you think? Is it like smell or what? Because there's not like a ton of sexual dimorphism for coloration or anything, right? I mean, I guess that's, you kind of look for a sexual dimorphism when you're looking for honest signals sometimes.
So you look for sexual dimorphism, but what you do get during the mating season is that the males have these tufts on their kind of on their shoulders nearby, and it has a pheromone and a musk that comes out of it, and that's clearly a signal.
Some honest musk.
Yeah, because like it only comes out during mating season for a lot of these male species. For some of the species, we don't know if they emit a musk, and so that's like a more difficult question. Especially because there's different dynamics with like colonial versus singular species and how they will interact. So most flying foxes, for instance, people think of are like these giant colonies, but there are a fair number of them that are only singletons or like coupled in the forest, and so they have a hard time finding each other, and then they like stay together, or they mate and then they separate. And those species, like we have no idea how they find each other, because like when we catch them, it's like, oh, by luck we caught one in our net.
Right.
Even we don't know how to catch them. There's no idea.
Also, just like the dynamics of musk in like a lek, it's like visual signals, it's easier. You can be like, oh yeah, that one for sure, like I want that guy. But with musk, you're kind of, it's like a scent trail that you'd be dealing, because like they're all flying, right? So that would be, that's hard.
They hang out at the roost, but it's like, I know their smell is really good. Like their scent smells great, but like that roost has a lot of bats in it.
Yeah, is it the difference between like that and like that? You know, you know what I mean? Like that's,
I feel like, especially with like a lek situation, you're left with like, you know where the center is, so the female should know where to go. And then they should be able to make a choice. But then what happens is, like, for the species that don't have leks but are still colonial, how do you find the males? And like for some of those species, the males approach the females, and so there's a lot of bat raping going on. There's a lot of like the males trying to force itself on the females, and that's actually why you see sexual dimorphism in some of the species, with the tibia being longer in the male, because then they can mount the female.
They can kind of hold on.
Yeah, and they can like hold on with less having to like close the distance, basically, right? So like males are a lot of the species that have this kind of system, like they have slightly longer tibias, but it's like mildly significant, you know, it's like tiny significance, and no one really knows like how effective that is in terms of like, is that really sexual dimorphism? Because there's not really a ton of other traits, and no one's really studied them. Like we don't have any idea about like calls or like social calls even that they have. So we have no idea how to like classify if they're making a sound that's like, okay, we're cool, versus like, oh well, get the fuck off me, right? So like we have no idea how to do that. Whereas with the insect bats, you know, you take echolocation calls, you know the approach calls, you know the buzzes, you know social calls, like you have all this other data because of echolocation. You have the meters with you, and you've been taking this data and you've been collecting the data.
Right.
And you have a reference for how it's going to be standardized, but no one studies that with these fruit bats, right? Because it's not the echolocation; it's regular sound, right? And so the soundscape is more like when you're dealing with birds, and you need to record it and analyze and try to figure out where things stop and start. But no one's really done that, and a lot of it is very screechy. So it's very hard to like get good data from a single bat. So, and you're not gonna keep like a flying fox and like a flight cage and try to get it to like call each other. They're just gonna be looking at each other like, this is dumb. Like, why am I in this cage with like one other guy? Cause I'm just gonna stay here. Like, so like they're just gonna look at the other bat like, whatever, man, I'm over here. So yeah, it's different in terms of the system, right? So you can't do the same thing. But they did show with a colony of Rosettis. So there's a lab in Israel that studies a lot of these call signals, especially for echolocation calls. But Rosettis are special because they're the ones that are these very early fruit bats that still have this like clicking system, and they happen to be a cave species, so like that's not a surprise to me. So they do these like really kind of crude clicks sometimes when they're flying instead of just using the whole sight and smell thing. And they once actually blacked out this entire lab in Thailand to show that like other fruit bats might also actually do that when it is totally completely dark and there are literally no other signals that they can depend on, is that they might also make a clap thing with their wing to
Oh,
mimic
cool.
the rosettas are giving off these clicks. And in that way, they will get like a sound coming back so that they can also like figure out where they're going. But there's almost no data on that because no one's gonna run an experiment like that normally. Like that's the only experiment we have right now, and that was only like 5 years ago, right?
Wow.
Right. So these guys in this Israeli lab, they did this really cool thing with rosettas. They have a colony in their lab, and they basically found that like the rosettas talk to each other. And when they talk to each other, they are also teaching each other the social calls. So the pups learn the social calls from the mothers that are in the same colony with them, their people, the other bats around them.
Yeah, it's like their culture.
They have a linguistic culture clearly, because
Yeah.
otherwise like they won't be able to social call each other. And they're in these really tight spaces. Like they're like thousands of them crammed in like pretty small areas and caves. So like clearly they're like just all on top of each other. And a lot of the bat pups, when you look at insect bats, like we know that a pipistrelle can call and the mother will come back to the roost and be able to find her pup out of the like thousands of them that are there. So there's very specific call culture and response and like understanding from the bats that are individualistic and also societal. And we don't know a lot about it because we've only really started studying that like recently. It's also like, genomics, where it's like, oh, now we can actually record sounds. It's like a whole different line of evidence that we like never thought about before. And so everyone's like, wow, we have all this data. And then we're like, no, we need to make a central repository. And everyone's like, we don't know how to do that yet. Because holding large media files is still a challenge for science.
Yeah. Yeah. Well, it's cool. It's cool that you're trying to solve that problem, I guess, because that is an important one.
No, that's why going to the bat meetings are always fun, because like you meet everybody, and it's like the fact that you can like absorb all this information and you don't have to read the paper. So it's great because like I am tweeting a lot of my bat conferences because one, it's a way to disseminate the information, but two, it's a way for me to also consolidate what I just heard and like have important points like typed up really quickly.
And then like you can look back at your feed and be like,
yeah, exactly. And like what I do with a lot of conferences, I make like a wakelet, like you have the whole story thing for the entire conference. So I will archive every single day. We have all the tweets from a single day from not just me, but everybody else that uses the tag for the conference. And that way you can go back and look at it later on if you need to. And I think we have like some really good speakers in our field in general. Most people have been trained to not just like talk from like a index card or whatever. And the mentors are usually very hands-on. So like you get practice with your mentor. So your talk is good, even if you're an undergrad.
That's so cool.
We also do this thing where the first day and the first 2 sessions, and now it's like first full day, it's only students.
Cool.
So basically all the students, if they want to, can check off that they're entered into the student honors like contest basically, right? The award stuff. And if you're a student that doesn't want to do that, that's fine. Like you can be with like the regular ones, but if you're in the competition, everything you're going to be compared to is only other students, and no PIs and other people would have talked at all because you're on day 1. And that way people also know, oh, you're a student, I should go talk to you, so that you don't feel like left out because you already like talked at the conference. So people know your face and know your name and know what you do, and you don't have to reintroduce yourself to like 400 people. Yeah. So I think that's good. And most conferences don't do that because they mix the students into the sessions based on themes. And that's just like, that's a losing battle, because then you have to order people in a certain way so that the junior folks always speak first, and that might not work thematically based on what the talks are. Right. So we still do that even within like a session. The chairs will order it so it's like the junior to the senior folks, or like the broad to the specific, depending on which sessions they are. And it's because of the fact that we all know each other so well that we kind of can figure out where to put people so that it makes it like less uncomfortable for people that are new and people that are first-time presenters, for instance. So yeah, so I think we do a really good job of making sure that students feel really welcome. And so we have a really good like retention rate of students from undergrad into graduate school.
Oh, that's
And now
awesome.
postdocs. So yeah, so I think our field is growing and our society is growing also compared to a lot of other societies, which I will not name specifics, but I know that they're shrinking because they have terrible policies or they have members who are like, you know, the old boys club kind of thing and no one wants to go to their meetings. So
Yeah.
yeah, that's part of why they suck, because no one wants to go to their meetings. When you look at like demographically, where are people at now? It's not their meetings, right? And that's also why like evolution has done a really good job. They have all these measures now that are very clear, and they're addressing a lot of things that students need for support, if or early career researchers even that need support for if there is an incident that happens to them at the conference. And like everyone who is a senior person who is helping with this is like really gung-ho about making sure it's actually like abided by. So like everybody is being pushed to use these resources, and it helps the meeting a lot, because I know when I went in 2010, I think. Yeah, I went in 2010. So that was the first summer after, it was after my first year of my PhD. And so it was like 10 years ago now. Damn, it was like 10 years ago. I'm like, wait, 2010 was like a decade ago. Holy crap. Time passes really fast.
I feel like 2 months ago was also a decade ago somehow.
That's
like
true.
February was also a decade ago. Yeah.
That was a lifetime ago. That's true.
Yeah.
But yeah, like it was like uncomfortable for a lot of people who were like young female researchers, and now it's like a really good meeting to go to. So it's
That's
definitely
awesome.
changed. Like, some meetings have really like tried to change, and they've done really well. But I think that meetings have always been very like we're like family almost, and like we get along with each other. So yeah, and I mean with the occasional hiccup of certain terrible people, but like we know who they are, and so like people can shun them.
Yeah,
it's fun. But yeah, no, I always encourage people to work on bats, because like there's tons of questions in every single possible field, because we haven't studied bats like a lot compared to other animals. And you can basically pick anywhere in the world to work in, and there's probably a bat species or species complex there that would be interesting and has interesting questions associated with them. So you know, I picked places that I was like, yeah, Southeast Asia sounds really fun.
That sounds
and,
cool.
Like, that sounds cool. There's like all these islands to go to, and some like the tropics. I love the tropics. And I'm like, oh, the reason why no one studies them is because they're in so many countries that you need permits for like 40 places. If you want to
it's hard.
Yep. And I have permits in 5, and it's already more than most people do, because like most people study one country's like flying foxes, and they're like, I'm done, because I don't want to get permits from like another country. And it's hard because they have CITES protection, so like you can't bring stuff across the border unless you're working with a CITES institution. Which fortunately, because I work with museums, they are CITES institutions, but most people don't, and so you need like an extra permit to export and import, and it makes it really difficult for them. And like when I started my PhD, I was like, this is a cool question, you know. They're bats, they're like in this island landscape, and they're really diverse. They're the most diverse genus in their family by a lot, and like, why is that, you know? And I just was like, this is a cool question. And Dave being an insect person was like, that is a cool question. And
sure, that'll be no problem.
Sure thing. And then Nancy, being totally like too senior to think anything is a problem, is like, I'm going to support whatever you want to do. So like, go for it. So both of my advisors were like, yeah, you'll figure it out. It'll be fine. And I'm like, oh man, we're sitting here in this goddamn office waiting for permits all the time. You're like, I see now why other people don't study this. I'm the only idiot who is doing this. So yeah.
Jeez.
First year of my PhD was a lot of people saying, you're not gonna be able to do it. You're gonna have to change your PhD halfway. And at the end, they're like, oh, you actually did it. That's kind of insane. So you're kind of insane. They just look at me like, okay.
Jeez. Well, do you want to quickly talk about why Pteropus is so diverse in insular Southeast Asia? Or we've been going, this was, you know, I said 15 minutes early on. That's not like, I don't have anything going on right now. So I'm overjoyed to listen to you talk at me about bats, and like, it's really fun.
You know me, I like talking bats all the time. Well, so I can mention how the paper ties in a little bit, right? So
sure.
The paper itself is focused on biogeography, but it has a backbone of the systematics and evolution work, because we needed to do an update of that before we can answer the question. And so I went out and did a lot of field work in Indonesia because we were filling in these gaps for sampling that were necessary to answer the evolutionary question and then move into the biogeographic question. So I'm really interested in them because when you have these large island groups and you have somehow animals on them as native assemblages, you don't really know how that happens, right? Because they can either, especially animals like bats that have like high mobility, because you can either have something come in and colonize the island, or you can have something come in and then radiate into multiple species. So when you're on these islands, you'll see like 4 or 5 different flying fox species. And it's not very clear if they all just came in separately because they could, or if the one of them came in and then they just like diversified into all the niches and nothing else fills because there's nothing there. So nobody knows, right? Because no one's collected them, no one's seen them, no one's really thought about these questions in relation to them for a long time. Mm-hmm. And the traditional ways of organizing them were only based on morphology. And the last time there was a morphological taxonomic revision was 1918 by Newt Anderson at the British Museum, and it was from stuff that people sent him back, like skins that were skinned in the field and then sent back to the British Museum. So they're not, they're not the greatest condition, right? They're like pickled and like their heads look kind of funny. They're missing all their, details, and like the fur is like kind of weird looking sometimes. So you can't tell anything about like musk except for notes that people already took from the field otherwise. Like there's tons of gaps in knowledge, right? Mm-hmm. And so a couple of years before I started my PhD, Nancy had a postdoc who was also interested in this question, and they tried to make a tree for it. And basically the tree uses a lot of genes that we would use for studying other bats that were fine, and in Pteropus they were not fine, and basically you cannot solve any sort of the polytomies that form on the phylogeny. And the reason for that was like, we didn't know why. And so I just went in and did this, and I was like, okay, well, we'll use some of those genes and we'll add some other ones that like introns that are faster, because I know there are going to be some of the species that I'm including that are very recently diverged because they were like subspecies that were split into 2 separate species. So I needed that like level of diversity in the genes I was working with, because you can't presuppose what you're going to get from a gene, but at the same time, you can at least that you get different levels of data for different levels of questions. So what happened was we found out, one, Pteropus are not very genomically diverse, like in general, which I should have just predicted that, but like we can't really tell sometimes. In the family Pteropodidae, they have this large chunk of their genome, which is line 1. And line 1 is like really, really diverse in most mammals. It's just completely deleted. They don't have it in the family. And someone I talked to recently has said to me it might be related to how they also deal with aging, because it's actually very highly relevant to like cancer research for line 1. And so there are other things that might have been why they lost it. And, a lot of flying animals have very conserved genomes as a way to just be like, hey, we need to cut down as many extra things out of our body as possible because we need to be like efficient. Right. So they have these really small, tight genomes that are not very diverse, which is weird because you look at other bat species that are that level of species richness, like, Myotis and other stuff, and you'll see, wow, they have like lots of differences in them that you can track easily. And we have these 65 species here that all look kind of alike. They're different in terms of their forearm size and sometimes the color. And that's about all for the most part. Mm-hmm. And then like everything comes down to like teeth, everything comes to skull features that are really difficult. And so everyone's like, yeah, those are all different species though, because we know they don't mate with each other. And these islands all clearly have different species. So how did that happen? Yeah. And clearly island isolation is a big part of it. And when you get these spaces where there's just nothing, you get isolation, you get genetic drift. Those are like, your like little basic stuff that happens with evolution that you already predicted. But then you also get this type of evolution called peripatric speciation. So normally you're thinking of like, vicariance, where there's like a population and like a mountain range comes through it. And so it splits the populations, and then time happens and they become A and B species. So when the mountain is eroded, now they can't like mate because they're 2 separate species. these bats don't really do that because they can fly, but a lot of them also don't choose to fly very far if they are on
Right.
island tropical paradises that have all the fruit they want. So then they just stay like locally where they are, and they're just like, this is great. I have no competitors. I have no other bat species if like two other ones to compete with, and they just feed at a different time of day, or we just fight over it because like that's fine, and fruits like us fighting over it, so that's fine, right? So these bats are just like, they're living their best life on these
Yeah.
islands, right?
Yeah.
They love it. But because of that, you basically had a core population that somehow got one on this island, and then it's like at the edge of that, species range for the original one. And suddenly they're just on this island doing their own thing. And eventually that just means that they're just gonna be a different species. So you get this isolation that helps push this divergence because nothing else is coming into there. And it's not a regular route that's traveled by the regular species, also because it's at the edge of that particular species range. And you have a ton of that when you go to islands because there's just nothing, next to you a lot of times when you're looking at it. And a lot of people were like predicting originally that these bats are probably more Asiatic because the continental side of Wallace's line that has more of these species are in Asia, and like the bigger ones are there. And a lot of people kind of thought like the bigger ones might have been where they came from, but clearly that's not the case. Like once we started getting genetics, then we could tell that they were not the root. But we also didn't know where the root was because we were getting this really weird result that was like Papua New Guinea plus maybe the Philippines. And there are like a couple of species that do have this weird track of like Papua New Guinea, Micronesia, and the Philippines. But these bats have only like 2 species that do that. So it's like weird as a root. And it also doesn't track with like, how did they get to Papua New Guinea in the first place? Like it doesn't make sense. So because of that, there were like multiple places they could have started. Nobody really knows. Papua New Guinea has like tons of species. They have like 7 flying fox species. And so everyone was like, no, I don't know. We have no idea. Because then there was just all these islands that all have like 4 or 5 species. And then there's like one big one for all the continentals. And then like they have one side one. And no one knows how that happened. And so fortunately, when we answered the evolution question, we were able to fill in these gaps for all the different species groups. So the flying foxes have been divided into these groups just based on morphological characteristics, behavioral characteristics, and other like indicators basically that we know. Like behavior is actually a pretty strong indicator for them. It's just we don't know a lot of that information.
Right.
But when you do know it for certain species and you look at the roost, you can tell, oh, this one likes to be more clumped together and these ones like to be spaced out, and they're not on the same tree branch. Like they don't like being next to each other, right? Or they will be in different levels of the tree. Like one species will take the lower level, one is on the higher level. And there's a lot of these context clues that are based on just going out and doing research, which is why fieldwork is really important. But no one knows that. Like most people who study phylogenetics are just borrowing tissue samples from museums, right? And they don't have these notes in there. And so they don't know that that's the context for it. These are definitely different species, but how do you explain that? So I and a lot of other people who have done a lot of field work had hypothesized certain species that were probably splits, or they were recognizable enough that you can tell that there were probably more species because they were actually separated. And we filled in so many of these gaps that we were able to just say, okay, well, this is the backbone tree that you then do these biogeographic analyses to say, where are they coming from? Because your root then is the rest of the all the other old world fruit bats. So I picked, I think, 6 or 7 species originally that were from other genera that are kind of close to the flying foxes. And we were trying to like root it with one of these. And I knew they were kind of close, but not quite, so that you would get a good, a good nesting of places. But on top of that, I also actually, they have a sister genera called the Cerodon, which is like flying foxes, but one of their molars is more square. Which is the dumbest way to explain it, but it's true. That is the difference between the 2 genera. And they are very, very clearly distinct. Like they have no overlap genetically in terms of their, their signals, and the polytomies don't exist for a Cerodon because there's only 4 or 5 species. They are extremely like endemic and specialist to the places where they exist. And because of that, there's no way that they're like flying all over the place like flying foxes are, and like overlapping with migration and dispersal in a way that prevents evolution from happening, basically, because of preventing isolation. But these guys are like, no, we just got to these weird islands and no one else is here, then we just stay here. And like, that's it, right? So there's these other bats that we rooted them with also as a way to show like, okay, this is definitely like, this is Pteropus, like the core in group, because we know that the sister taxa, the sister genus is right there, and we like actually sampled a bunch of them. So it's not just like a artifact of what your sampling was doing, because it's not just one species representing the entire genus, it's 4 out of the 5. So, you know, robust data sets in terms of what we're doing.
Gotcha.
And that's always been the criticism for a lot of work phylogenetically on pteropods, because the sampling is really bad, because you have like 200 species and they're all over the all over the Paleotropics, and that's like, you know, Africa through Southeast Asia, through Australia, and then all the way out to like Fiji and Tonga. So like it's a huge breadth for a family, right? And when you look at that breadth, the only other bat family that has the same distribution is like Embolonura bats. Like Myotis bats, you know, like the really common ones that you see, they're everywhere, sure. But then when you're cosmopolitan, it means you can just be everywhere. But when you have like this very specific distribution, which is like just the old world tropics, only certain species do that. And it becomes a very interesting question to tackle, but also very difficult to tackle as a group. And so they've been the taxonomic headaches for a lot of people for a long time. And everybody who gets the question for like one of these genera or large sections of these families, it's basically an entire PhD thesis because of that, and has not been addressed in most of the other genera too. So when we looked at this biogeographic question, now that we have this tree that we are fairly confident in, because I ran way too many species trees and all these other things to it. This is like this is the part where you're working when something's like running on the computer. And you're
CPU hours equals
It's
Susan is
like
working.
a KCD comic where they're like sword fighting on chairs, but they're, you know, it's like compiling, so you're still working. So it's totally working. So we have a lot of information about the distributions, because we actually know where most of these species are presently, but also like from historically their distributions. So we did include a couple of the islands where like they might not be found now, but they're important, because when you have this crossover between the different regions, then you need to actually mark them as being able to cross that boundary. So we separated our land masses into geologically clear places. So, you know,
let me see if I can share screen so if people watch this video, they can actually see what you're talking about.
So we have a map in our paper. And it's the base of it is Dave's, you know, map from, like, I don't know, he likes playing with maps in Illustrator sometimes. So he had all these vectors and he made a really nice map.
Oh, I have to grant a ton of permissions to Zoom. This might be more trouble than it's worth. Maybe I'll just link to the paper.
It
might take me a little bit too long to share my screen. Sorry.
I have the same problem the first time I use Zoom, so I've had that already. So if you look at that figure, it delineates these big biogeographic regions instead of like little islands like most people are studying. These we're trying to look through this more global view of the genus. And so we have Wallace's line here. So you have Wallacea here, you have, you know, Asia here, and then you have Lydecker's line here. So Wallacea is the middle part in between those two lines. And you have like Micronesia, and then you have like the Pacific Islands over here, and then you have some Western Indian Ocean islands, right? So you have 6,
like Madagascar and the Seychelles and stuff.
Madagascar, the Seychelles, Mauritius, Reunion, Rodriguez, like all those little
guys.
Going on there. And Temba also.
I don't know that one.
It's in Tanzania, and there is one species found on Pemba.
No way.
And it's on Pemba, and like this, and then this little like peninsula of another island next to it, and that's it. It doesn't make it to the mainland, basically, which is no surprise, because flying foxes are just like, we can't compete with things bigger than us. So like,
so is there another pteropid there, or is
there are other smaller pteropods? So like, if you are looking at other like Eidolon or other more general species, they're on the continent. You know, they're always gonna be on the continent. You're gonna find an Epimorphus. You're gonna find all these other bats are pretty much the known assemblage fauna for continental Africa.
Mm-hmm.
And the flying foxes don't cross over. They just clearly, can't do it. And so they just make it to the islands. And I can talk about that later too, as to why I think that is. But when we're looking at the analyses, we find that, so our route ends up being Wallacea. And part of that might be a function of the fact that our outgroup, the sister taxa, the Ceratodon, is also actually a Wallacean genus. Not all the species are found in Wallacea, but clearly their origin was also Wallacea.
Mm-hmm.
And so when you look at the route before that, you get an origin that is like clearly Wallacean, because all the other bat genera are a mix of these other places like Asia, Philippines, Wallacea. So you have to look at probability-wise which one is more likely, and if your two groups are coming out to be Wallacean, then you get Wallacea. So we had a very clear, like, there is a Wallacean origin that is very likely. And there was like one point where there was like a borderline, it could be the Pacific Islands also, but they actually mean like Papua New Guinea, not like the Pacific Islands really. And that was probably a function of the fact that there was just one really large radiation. That is why you find like 7 species in Papua New Guinea.
Yeah.
But we found this origin in Wallacea, which is really cool, because that means that, one, they came from where the central part of their distribution is right now. They did come from that. It did radiate outward from there.
That's nice.
And that's always nice, because then you're like, great, the easiest answer was the answer for that part. And not these like weird, like it was on this edge part and then they like diversified everywhere, because that would be really dumb. So like the center part is the center part. That's always great. But then the center part that's the center part is a bunch of islands. And Wallacea has never been connected to any of the continents, which is the important part, because everyone's like, oh, is it an Asiatic fauna or is it an Australasian fauna, right? It's neither. It's Wallacea. Yeah. So it's just weird, because Wallacea is made up of volcanic islands. It's made up of pieces of other places that have just been flung off into there. Yeah. It's made up of the edge of continental plates. They curl up sometimes, and they have these things called crustals that come up and then form these larger stretches, but larger, not very large. And so you have all these islands that are separated, much like your lovely background. That is like the backdrop of a lot of the places these bats like to hang out in also. Yeah. Right. So like that, there happen to be a lot of spaces like that, and the bats will find somewhere to go because nothing else is there, and nothing bothers them there because it's so remote. So they get to diversify species, whatever. And that's weird, because most people think of an environment where the origin normally is like stable, quote unquote, like that, because they're thinking about vicariants mostly.
Yeah.
And so when you think about vicariants, you're thinking of a central like area that's splitting off somehow, because you're not getting this splitting off, and instead you are getting this diversification because these islands, land masses are coming out. Then you're just going to get colonization. But when you get colonization, that also means that you're not doing vicariants, because you couldn't have done vicariants because you didn't have continents. Like you have like maybe localized vicariants because like rivers or something, but you're not getting like vicariants on a broader scale, which is what people think about. So, if you look at South America, a lot of people think about the Andean orogeny. Mm-hmm. And when the uplift happened, you see birds, for instance, that are found on both sides of the Andean orogeny. And as it came up, you see that those populations start to diverge. And you see that the birds that are more able to fly higher, they diverge a little later, because that orogeny is longer to come up. So
in class, we talk about shrimps that live at different depths on either side of the Panama Isthmus. So it's the same. It's the same concept. Yeah. Just so, for any students that are still watching this video, just to tie it in.
So there's a lot of this really cool stuff that happens where it wouldn't happen in this case, because you don't have continents, you don't have these like stable places. You have continuous volcanism, you have continuous land movement.
Yeah. But it's like it's old-ish though, right? It's like 30 million years old, something like that. Like Wallacea as a potentially viable.
Different parts of it are different.
Yeah. But if you're, if you're a volant organism, that
So
sort of general fuzzy area is
yeah, so like
potentially
that
habitable for a long
area,
time.
the whole like Sulawesi part coming down like that.
Yeah.
There are parts of it that are coming out, and then there's like this submergence back and forth, but that doesn't really matter right now. It's just like there is a landmass that's moving somewhere, and then
Yeah.
there's these little islands popping out. But you get that Lesser Sunda thing.
Hmm.
Those are sometimes they're moving in from other places, or that they're volcanic islands that are
Yeah.
coming in pieces, or they're just sloughing off some other islands, and they're just really
Right.
strange how they appear. And
Yeah.
so, and once I remember I had to look up how the Moluccan Islands were formed, because Maluku is very strange. Also, it has a very obvious, Saram is really big and forms as like upside down C kind of form, and then there's like
Hmm.
3 other islands near it, and they are a very close knit complex of islands. And then like right above them, equidistant really, there's like the North Moluccan Islands that are like they're a big unit themselves also, and like these animals don't want to cross this little bit of ocean that is the same distance between the 3 islands within the same unit.
Yeah.
And you get really strong biogeographic barriers that we can't explain very well, because we don't know things like wind condition or other currents that can also impact if animals will regularly cross those types of boundaries. And we don't know enough from some of those periods yet, because the data doesn't exist, right? So a
Yeah.
lot of people who work on this question of like Sulawesi, Sulawesi is 4 different landmasses put together, and how and when those happen have always been like a major question, because it's such a big landmass in Malaysia. And I remember when I started my PhD, everyone was like, okay, the 4 of them came together, and then it was like Voltron, and it formed Sulawesi. And then by the end of my PhD, Robert Hall gave us a very long and important plenary talk where he was like, my mistake, our data now shows that there are parts of it that are actually expanding outwards. And so you're like, oh my God, everybody is just like, didn't we talk about
Scrambling
this?
to rewrite their
Yeah.
discussion sections. Yeah.
I mean, and it's funny because we already kind of knew that our hypotheses were not very dependent on if it's expanding or colliding, but at the
Yeah.
same time, you do want to know, because that means how much closer are you to the other land masses in the South Pacific or in Micronesia
For
or
sure.
Philippines, and
Yeah.
suddenly you're like, oh, okay, like this
Yeah.
is very different from where we thought we would be, because that also changes which parts of Sulawesi are submerged, which parts are not submerged.
Right.
And so those are still like things that are happening in research. They have places they want to target in Sulawesi to sample the cores and to sample the rock, so they can actually get data, because they haven't sampled there yet. And these guys get like oil money to go on a boat to all these little islands and like get data most of the time.
Yeah.
And they still haven't sampled all of Sulawesi to a sufficient degree where they can reconstruct every part of what's happening. So it's a really weird question about what is the true story kind of Sulawesi sometimes. We
Yeah.
have a best guess right now. But yeah, every other island is like really tiny, and everyone's doing like their own thing.
Hmm.
And so when you get these batches popping out of there, eventually one of them will just pop off to like Asia, and one of them will like pop off to Australasia, and one of them will pop off to like Micronesia. And you're just like, okay. But instead what happens is you have the group that goes off to Asia, and those are the ones that people think about most of the time. They're really, really big flying foxes. They're colonial. They are the ones that people think of as flying foxes because of the fact that they are big, right? So there's like one species on each big continental landmass. So you have like Vampyrus is the one that's in Southeast Asia. You have Giganteus is the one that's in India. You have like Rufus that's in Madagascar, right? So everybody has one, and then there's some in between islands.
Mm.
And then Vampyrus goes into the Philippines, and it makes a sister taxa, and then that somehow also goes into like Micronesia, and it's just like a lot of weird things happen because now you have these different routes. But behaviorally, when you look at them, they are actually species that are also more dispersal prone. So they actually are the ones that are flying like 50 kilometers to go and like find some fruit in Malaysia, even though they live in Indonesia, right? They're the ones making these daily trips across the Strait of Malacca just to go find some good fruit.
Yeah.
Then you
Wow.
get these other guys on the islands that I'm working on in Malaysia where they're like, I'm going to hang out on this island.
Yeah.
That's it. And I was able to catch them because we put a net in the mangrove, not even like in the forest. Like it's like really insane where sometimes you find these bats, and it's very difficult to catch them because of that. But I always tell everybody I always tell the grant people, I'm like, they're really easy to catch because I've been doing this for a long time now.
Yeah, I got it. Don't worry about it. That's fine.
So long, it's really easy.
This grant is plausible.
It's plausible to me.
Yeah.
So hilariously, most people are thinking like, oh, they're like super dispersers, and so that's why this, when you have island separating, it doesn't really matter. Mm-hmm. No, they're really lazy actually. So like, you get to a good island, you're just gonna just do nothing, right?
Yeah.
So that's what happens. Like, they do nothing, and it ends up being these really interesting species engines where you get some cases where they are colonizing islands multiple times in order to reach land masses.
Yeah.
And then on some land masses that are more difficult to reach, you get one that gets there and then it radiates. Mm-hmm. So you see that only in the Pacific Island one. There's a radiation that goes out from Papua New Guinea and it goes all the way out to Samoa and Tonga. So every single Micronesian, Melanesian island has their own like species. Every Micronesian one also does, but it's from a different, it's from a different radiation. And most of the time when you think about island biogeography, this is the mainland, and everything that is further from the mainland is going to be less and less and less related to what was on the mainland, because it's just isolation by distance.
Kind of a stepping stone kind of model.
Right. But they know the bats are lazy, so like they still think like stepping stones probably is what happened. But no, you don't get that in Wallacea. These bats just go all over the place because everything is full of food. There's literally nothing else on it. So they're like, great, I'm gonna eat these figs here because I love figs, and figs also happen to be an early succession fruit that you need to break up rock and soil and other stuff. There's no other things to eat. Figs are small. They have small seeds, and bats have tiny esophaguses, so they need to be able to like mash it up and swallow the pulp sometimes. Yeah, figs are perfect. And then sometimes they have wasps in them so that you get some protein also out of it. It's great. Right. So these flying foxes are just having the time of their lives in Wallacea. But then once you get to the Pacific, where you do have more limited islands to pick from, then you basically have Papua New Guinea acting as a continent origin. Right. And isolation by distance happens as you go further out. And so they island hop only for these Pacific ones. And it's really interesting to me because I've started reading a lot of books that are about people who study wayfinders in Pacific cultural anthropology, because we've been getting more and more information about that in the past like 10, 15 years that are accurate from, including people who are from those places and their narratives, because then you can figure out where they went, and where they went often tracks with sometimes, where the currents go, but also because they're able to, direct their own dispersal. The ways that you look at where the winds are blowing are usually how do we get back home, actually. Because if you adventure out somewhere and you get lost, at least if you can find that current under that star that it will bring you home. And when you look at the direction that some of these bats are dispersing, it follows some of these wind currents that are like congruent with where you're finding people also like going back outwards. And it's pretty indicative, I think, of how much they are dependent on these environmental conditions to be able to disperse that far in ways that might not be easy, because you're trying to hit a very small landmass in a very large ocean. And that's like 1 in a million for an animal sometimes, because you don't know what's out there. Right. So it's really weird because of that. And all of this happens in a very, very short span of time too. So you get some of these radiations are like 2 million years old. Some of them are like 7, but basically the root with the sister genus is at maximum like 14 million years ago. But it's probably closer to like 10 to 7, honestly. And the genes start diverging earlier. When you have that time period, you're already pretty much in a very late era, compared to other bats that we think of, and other bats that are at that level of like species richness. And you get this huge explosion of them in all these different spots. So when you actually calculate the evolutionary speciation rates with like BAM, you get this huge spike in them. And it's something like, I did this in my thesis, and we haven't written the paper for it because like the data was thin, since I don't have like a ton of other species to compare it to. But it shows that there is a huge spike in Pteropus where it's like ×6 increase in
Oh,
their
wow, cool.
speciation rate compared to other pteropodids.
Hmm.
And then when Nate Upham's paper came out, so that there was a big mammal tree, and he
Yeah.
did like BAM rates on a bunch of them. They found them mostly in species that were like rodents, for instance, where they'll get somewhere and they get isolated and they have lots of babies, and so like they speciate, right? But
Right.
like this is the only highly mobile species that for whatever reason has all this stuff happening to it, and they can't explain it very well. And it's only in this genus out of that family too.
But Wallacea, it's just such.
It's those islands, yeah. I'm
Yeah.
with you. Those islands, you get isolated in a different way, and so you're just stuck doing this.
Mm.
And these bats are so weirdly diverse in a way where they don't have to change morphologically because all the fruits are basically the same. You just mash it up and you drink it and eat it, and the plants don't care because if you look at how fruit guilds evolve, it's not like with pollinator guilds where they have to like match the corolla to the bat.
Right.
You just need to have enough frugivores in that island's assemblage that can handle your fruit and then disperse it.
Yeah.
And it's pretty agnostic in terms of the plants like trying to target stuff because of that sometimes.
Yeah.
So it's just who is the better disperser at that point. And bats are better dispersers than birds in some of these cases. But the plants are just like, whatever is dispersing me, I don't care. Like it will work, right? So there are fruit doves that are in the South Pacific that were also serving a similar purpose, historically. A lot of these fruit doves in some islands are now extinct because people ate them and they are like gone now. So all these Ducula fruit doves gone, and the fruit bats are the only native frugivore that's left on these islands.
Mm-hmm.
So they are literally the only thing making sure that there's a forest still there because they can actually cross these distances between the islands and connect the genetic community between the plants that are there. So it ends up being this really like strange idea of just like, okay, so these bats are so mobile that they basically do all the things that people think they can do, but like in different specific clades. And then also they are like super diverse compared to all these other bats because they're just like, they ramped up their evolution really quickly once they came to be because there were all these empty islands and everyone just like went nuts.
Yeah.
And Wallacea is really distinct and unique on the earth because there's no other place where it's just like, this is a tropical zone with places that are separated because most of your tropical zones are on continents. And so when you're thinking about that species engine in other continental areas, you have to depend on other ways to separate your populations, like rivers for some animals or mountains or, oceans. And you don't have that here. You just have like everything just popped out and it was empty. So we're just going to go like, that's fine. Yeah. Right. So it's really cool because we don't really know the answer for that, but that's our best hypothesis right now. And I don't think people really want to try to answer that question that much again because it is like a difficult thing to do. But now that we have all this tissue, I always try to get people like, hey, you should do all the genomes for these so we can make it like an even better tree. I put that paper in review, actually, that's for that.
Cool.
It's for like all pteropods, and Nico, a colleague of mine who's a postdoc in the UK, kind of dragged a bunch of us into this, and we name a new genus in it. Well, I name a new genus in it also. Yeah, there's a whole thing.
Oh, cool. That's awesome.
Yeah, there's a lot of stuff that happened in that paper where we're like, man, this took like way too much work. Like a lot of work, and he didn't have to do any of the field work because he just dragged like 12 of us into this and said like, do you have tissue samples? And can we do the genomics for it?
All right, man. Yeah.
He's a brave man for his postdoc to try to do that. And he didn't even capture all like 200. He got like close to like 110, I think, and it represented every single genus. Okay. So it looked pretty good already. But we can't answer these questions also because if you look at Pteropus, there's 65 species, and then every other group, the most diverse fruit bat groups otherwise, like Nyctamini, which are the nectarivorous like Yoda bats that are in the South Pacific Islands and parts of Indonesia, they are small, but they are also prone to doing a lot of this dispersal and being able to be generalists and sometimes specialists, but they are also like cave dependent. And some of the species are very tied to if you have caves to actually roost in. So they are more limited, right? I'm sorry, and that's Dobsonia, sorry, I'm thinking of a different bat. But like the Yoda bats are the same thing. So Nyctamini and Dobsonia are these other big groups, but by big, they mean like 10 species. And and you don't get this like huge explosion of species like Pteropus does. So you actually end up getting similar biogeographic patterns, but other limiters on them, not only by size, but other ecological factors that are probably making it difficult for those bats to exploit as many spaces as they can, even though
like maybe they're more like roost site limited or something,
it could
or
be like roost site limited. It could just be, Nyctomini are like tiny, like they're like that big. And
Gotcha.
then like Dobsonia are like medium size. And then that's the smallest size for a Pteropus, right? So like Pteropus can be like that big, right? They're like, their body sizes are long. They're like my forearm, right?
Right.
They're big, and you don't have that. Even if you count the smallest Pteropus species, it's the size of a medium-sized bat. So you're not going to get the same dispersal capability sometimes, even though it is a generalist that can like cross over these spaces. Especially for Nyctomini, they're like forest dwellers. They're kind of delicate also. Their wings have these like cute little mottled patterns and stuff. And you're like, yeah, you're not going anywhere. You're just going to be on for the rest of your
Just
life.
stay there and look pretty.
There is one called Nyctomini Hobbit, because it literally is just like on this one mountain in Papua New Guinea. That's the only place you find them.
Oh, that's cute.
They're just very strange bats. And yeah, there's tons of stuff with them where we just don't know enough, but there are probably limiters like ecologically that we are guessing is why they are not found as broadly as Pteropus. But they can be limited more by like elevation also. So if you actually have Papua New Guinea and you go up these elevations, you get fewer and fewer fruit bat species as you go up the elevation. But from the lowlands, you get more of the Nyctomini, for instance, and the other pteropods, and you get fewer of the other larger Pteropus, because they only stay on the lowlands. And as you go up, you get another species because the plants change for the Nyctomini. And then when you go up to the lower to mid part of the mountain where the plants change again, you get a different Nyctomini there. And then you get one on the top because they don't go that far up. But you get these really cool divisions because of altitude also.
That's cool. Yeah. So for students, Papua New Guinea, pretty big island, and there's a bunch of mountains on it that are more than 4,000 meters, right? Like there's several peaks that are above 12,000 feet.
There are 3 major mountain ranges in Papua New Guinea that are the biogeographic headaches for a lot of people.
Yeah. They're really hard to get to too. You have to be like...
Oh, you have to get a gold mining company to helicopter you into some of those places to stop. And that's what the AMNH did at one point. So
really? Oh, that's awesome. Was that like recently or?
This was
like in the 1800s?
No, this was 3 or 4 years ago. This is like recent. Yeah. So there's a special fund in the AMNH called like the Explore 21 grant. And it's like a bunch of trustees throw a bunch of money into it, and they want the curators to do interdisciplinary work. So all the vertebrate zoology curators are like, let's put a big project in for Papua New Guinea. And that counts as different departments, as it's like ornithology, mammalogy, etc. And then they threw in some.
As an invertebrate person, I'm outraged by that.
No, we had invert people too.
Okay.
We had a person who did microbiology and an invert person. So they put.
So you got the majority of biodiversity covered.
Yeah, you got a majority of the stuff that we needed to. We just excluded, gemstones and astronomy over there in physics.
Sorry, astronomy.
No one cares about that. So it was really funny because we were competing against one that was, I think, astrophysics and like cultural anthropology or something else. Basically, ours just looked cooler, and the trustees like have some money. And so we were really lucky that there was a postdoc who just came back from working in Papua New Guinea. So he knows the permit system, and he knows people on the ground that we can work with. And he has graduate students who can come with us on the expedition also to help. And so we were like, oh, this is great, because then we have like a local setup actually, and we don't have to like start everything from scratch.
Yeah.
And part of the money had to be spent on, because the original way this was made was there would be 4 elevations that we would be sampling at. So like lowland, low mountain, mid mountain, high mountain. And what happened was they said, well, to get to the high mountain part, you actually might need to get like a helicopter to fly you in between some of these places, because those peaks are not reachable unless you use them. And that didn't happen because basically it rained too much, and so they had to change the way that they sample elevationally. But you still need the helicopters or the planes to get between different parts of the sites, because we had 4 separate sites also. Right. So they still had to fly a plane, and you're like, man, these islands are so difficult. Like these sky islands are difficult to get to. Like
Yeah.
they're impossible to get to from the ground sometimes. You cannot go through the amount of brush and trees and rocks that's there. Like it's impossible.
It would take like a month or something.
It would take a month, but also it's raining, and so the ground is slippery.
And
So
there's an
it's
80% chance you would die. Yeah.
it's just gonna wash you out. There's a great video of them. They're under a tent and they're processing the specimens to get like tissue samples and like get the skins happening. And it's raining so hard outside you can hear it on the video. And a river is running through the camp. And it happens to be in a ditch that goes right under the table where they're doing the processing. So everyone is sitting there with their legs crossed on the bench because they want to get the water on them. And the water is maybe like 2 inches away from that. And they're just like, this is the way we have to sit like all day because we don't want to just put our feet in that water because it's
Hope
really
it doesn't get higher. Yeah.
Yeah, it's like rushing like crazy, and you can hear like Paul Speed going like, well, that's going pretty fast like in the background. It was crazy. Like that expedition, I almost went on the expedition and I still have regrets that I couldn't go.
Yeah.
Because that was the last year of my PhD.
Oh, yeah. You got to pick your battles.
And originally Nancy told me to go with them. So I helped them with parts of the proposal also. And I gave them my paperwork even. And then about halfway through the process we were doing this, Nancy's like, oh, wait, you have to graduate this year, huh? And I'm like, yeah. And she's like, you should write your thesis instead. My own advisor forgot that I had to graduate that year. And we were both like, Papua New Guinea. And then we were like, oh yeah, you got to graduate, hon. You really want to graduate, get the hell out of here. And I'm like, yes. So she remembered at the last minute, and she's like, yeah, maybe don't do that. They weren't traveling in the summer like we normally do. They were traveling like in January, February or something during the school year where I should be writing before I defend today. And it was way too close. So unfortunately I didn't get to go on that trip, but we have really great specimens from that trip that are very recent.
That's awesome.
You should totally talk to people who went on that trip too, because they deposited most things I think already for that particular expedition. Or they're like,
there's already someone who's kind of like Mr. Ants of New Guinea. I bet he's already kind of snatched them up.
That may be true, but I feel like there were so many things going on there that I don't know if he talked to all the AMH people, because he might have talked to people when it wasn't a session. So it might be worth a shot. But also like even just talking to like Dave General in the Philippines and stuff.
Oh yeah, hell yeah. No, I for sure. Yeah, so my advisor, we actually tried to get in touch with him. He like was, I think he was kind of MIA. He might have been on an expedition or something, or I don't know. But
he's been on a lot of them because he defended his master's and then he like started going around a lot of places.
Yeah. So I was able to meet up with him after the Indonesia thing, and he was super duper nice and showed me around and introduced me to people. But then we couldn't quite get in touch with him when my advisor was going over to the Philippines, but she was able to make some other contacts over there. And
he's around a lot because he works with Arvin and them. But do you meet Perry at the National Museum of the Philippines?
I don't think so. No.
So Perry is the entomology guy in zoology now in the National Museum. He's kind of younger because he replaced the previous guy that Dave used to work with that Dave also disliked a lot. So Dave was like, great, someone who's like younger and more knowledgeable and actually helpful in the field is going to come.
Oh, maybe I. What's his last name?
Buenavente.
I think maybe I did meet him. Yeah.
He's really young. He's got like a kind of mixture of Filipino and Chinese ethnic features. He's got glasses and he's got kind of like a faux hawk.
This dude wasn't rocking a faux hawk, but I did meet a younger person who seemed to be kind of new, and he seemed to be really knowledgeable about entomology and stuff.
So that might have been
maybe switch up his haircut.
Yeah, because he has kids now, so he has a different haircut.
Punk days are over. Yeah.
It's like there's like gum in his hair sometimes. But yeah, he's there, and he's actually really good to work with because he also really likes ants. And he is super friendly. And
I must have met. This must be the dude. I'm just a terrible person because I forgot his name. Yeah.
Because Dave also has him as his counterpart now for the National Museum. But Perry is really great because he can actually put you then in touch with Dave and Aral because they work together a lot.
Yeah.
And he knows all these guys who are at UPLB like Dave was because they work a lot with the scientists that are there.
Mm-hmm.
So they should be able to connect you with that. but they also have a connection to the students that are at Ateneo, which is in Manila. And they know a lot of people in Diliman just because, they are also a very large natural history University of the Philippines school. So that's in the south. It's on Bacolod. So I think that's Negros Occidental. It's on one of the Visayan
So
islands.
one of the Visayas, yeah, cool.
So it's a really good school for like
Cool.
Silicon, and then there's Diliman. There's a bunch of like really good schools that are around there. Diliman is like right there. There's Diliman UPLB, and there's Silliman and all these other ones that are in the Visayas that are really good also.
Nice.
They produce a lot of conservation biologists that now work on specific community conservation projects to protect species that are endemic to these islands in the Visayas and in Mindanao. So there's a lot of really good work being done down there. And they're often people that work on things that people don't care about, like snakes, frogs, lizards, inverts,
Yeah,
right? So a
that's
lot of
really cool.
really good people to work with.
That's awesome. Well, Susan, I think I'm gonna cut you off now
Yes,
because I think, yeah, I just need to,
few
I
other.
don't want to, well, actually, I don't have that much else going on. But I do want, no, but I do, I want to make this video somewhat manageable for the students and everything.
No,
But thank you so much for doing this. This was really awesome. It was really great to talk to you. And
yeah, no, really good catching up also.
yeah, yeah. So I hope we run into each other at some point, you know.
Yeah, if you're down in New York at AMNH once we're allowed to like be in museums again, just let me know.
Yeah. I was gonna go. There was like an ant meeting or a social insects meeting that was gonna be in like March, and we called it off. But
Yep,
that might get rescheduled at some point, or yeah,
I
it'll
remember
be
that.
something. Yeah,
Yeah, definitely
for
we can see
sure.
it in the email. So yep, yeah. I'm back
Yeah,
a lot between DC and New York. So yeah, just let me know.
awesome. Yeah. Well, stay safe, and yeah, thanks so much for this. This is
Yep.
awesome.
See ya.
Yeah, see you, Susan.